So these cells can communicate with one another. For example, maybe this cell sends out some kind of signal, releases some kind of molecule. This molecule can bind to a receptor on this receiving cell. And when it binds to the receptor goes through some kind of conformational change. When it goes through a conformational change, it can trigger a cascade of events, eventually activating other cellular proteins. And these activated cellular proteins can cause some real cellular changes within the cell. For example, maybe we activated a transcription factor. Now the transcription factor can enter the nucleus. When it enters the nucleus, it can activate the expression of certain genes, creating certain proteins, which can cause some real cellular changes. So this process is referred to as a signal transduction cascade, because this signal has been transduced within the cell. So what are some common examples of these signal transduction cascades? Well, the most common example involves these G-coupled protein receptors. So these G-coupled protein receptors normally have these 7 transmembrane domains. And they're called G-coupled protein receptors because they're bounded to these G proteins. And these G proteins, you can see, have these hydrophobic domains that allow it to latch onto this plasma membrane. And again, these G protein receptors usually have 3 subunits: an alpha subunit, a gamma subunit, and a beta subunit. But normally these G proteins are bounded to essentially a GDP. Normally this alpha subunit is bounded to a GDP molecule. And when the alpha subunit is bounded to a GDP molecule, it's off. It's turned off. However, what can happen is this signaling molecule can bind to the G-coupled protein receptor. When it binds to the G-coupled protein receptor, the G-coupled protein receptor goes through a conformational change. When it goes through a conformational change, it reacts with the G protein. And when it reacts with the G protein, it allows this GDP to fall off. And when the GDP falls off, a brand new GDP molecule binds. So now a brand new GTP molecule has bounded. And now when this GTP has bounded to the alpha subunit, now the alpha subunit has been switched on. And now the alpha subunit has been turned on. And once it's been turned on, essentially what happens is it dissociates with this gamma and beta subunit. And this gamma and beta subunit can also do some interesting things, but in this video I want to focus on this activated alpha subunit. So what does this activated alpha subunit do? Well, again, we know it has this hydrophobic domain, so it can diffuse freely within the plasma membrane. And normally what happens is it will diffuse and activate this adenyl cyclase. So it'll diffuse and activate this adenyl cyclase. When it activates this adenyl cyclase, essentially what it does is it takes an ATP molecule and converts it to a cyclic AMP molecule. So this activated alpha subunit activates this adenyl cyclase, which allows it to take an ATP and convert it to the cyclic AMP. And now the cyclic AMP molecule can cause some changes within the cell. And just a little bit of terminology, this molecule is referred to as the primary messenger. This cyclic AMP is referred to as the secondary messenger, and this adenyl cyclase is referred to as the effector. And the effector is what creates the secondary messenger. By definition, the effector is what creates the secondary messenger. And the reason why they're called this is the primary messenger was the original extracellular signal. So this original extracellular signal, this primary messenger, essentially activates this cascade of events, which activates the secondary messenger, which is the intracellular signal. So it's kind of like the secondary messenger is completing the work of this primary messenger. So the primary messenger binds extracellularly, causes changes that creates this intracellular secondary messenger, which can complete the changes inside the cell. And again, the effector is by definition what created the secondary messenger. But the point is we create the cyclic AMP. And now the cyclic AMP can cause some real changes within the cell. For example, normally we have this inactivated protein kinase A. So this is a kinase. So it's a protein kinase A. So it's a kinase, so it can phosphorylate other molecules, causes changes. However, normally the protein kinase A is inactivated. It has this yellow portion that's inactivating this protein kinase A. However, once we form the cyclic AMP can bind to this yellow domain. And when it binds to the yellow domain, it essentially dissociates this yellow domain. So now the protein kinase A is activated. So now that's activated, now it can act as a kinase. And it can phosphorylate regulatory proteins, or it can phosphorylate enzymes or transcription factors, either activating or inactivating these regulatory proteins and enzymes and transcription factors. But the point is, it will cause these real substantial cellular changes within the cell. So again, This may seem very arbitrary, it may seem like a lot of very specific and random steps. However, this is the most famous signal transduction cascade that you'll see in a biochemistry class or a cellular biology class. So I really recommend you memorize these steps because you'll see the signal transduction cascade over and over again. But again, this is just one example of a signal transduction cascade. For example, maybe let's say we're in a completely different cell with this G-coupled protein receptor and this G protein. We know what happens. This signal binds, causes a conformational change, and allows the GDP to fall off. And when the GDP falls off, now a brand new GTP can bind to the alpha subunit, activating the alpha subunit. So now we have this activated alpha subunit. And we know it can diffuse activating effectors. And another effector it can activate is this phospholipase C. So this activated alpha subunit will bind and activate the phospholipase C. And now that it's activated, it can act as a lipase. We see it's a phospholipase C, so it's a lipase. And once this activated alpha subunit activates this phospholipase C, now it's activated and it can create a secondary messenger. So how does it do this? Well, we know in this plasma membrane there are a lot of phospholipids, and one common phospholipid you'll see is this PIP2. So essentially what happens is once this phospholipase C is activated, it'll take this PIP2 and it'll act as a lipase. This phospholipase will act as a lipase, essentially hydrolyzing this phosphoester bond. And when it hydrolyzes this phosphoester bond of this PIP2, it essentially creates this DAG and this IP3. So this activated phospholipase C hydrolyzes this bond, creating a DAG and this PIP3. And now this PIP3 can cause some real changes within the cell. And again, just a little terminology. This was the primary messenger. This was the secondary messenger to complete the signal, the message. And again, this phospholipase C was the effector because it was what created the secondary messenger. And that by definition is what the effector is. So now that we created this PIP3, what is it going to do? Well, essentially what it does is it binds to this channel on the endoplasmic reticulum. So it binds to this channel and it opens the channel. And now that this channel is opened, it opens this calcium channel, allowing calcium ions to flow inside the cytoplasm. So again, this PIP3 essentially opens this calcium channel, allowing calcium ions to enter inside the cytoplasm. Now the calcium ions will bind to calmodulin. So when the calcium ions bind to calmodulin, now calmodulin is activated. And now that it's activated, it can cause real cellular changes within the cell. It can have some kind of cellular effect. But not just that, remember this DAG? This DAG can also do some important things. For example, this DAG can bind and activate this protein kinase C. So this protein kinase C binds to this DAG, and when it binds, now the protein kinase C is activated. So now that it's activated, it can act as a kinase. Now that it acts as a kinase, it will phosphorylate proteins and structural proteins and enzymes and transcription factors, causing a real cellular change within the cell. So these are just a couple examples of these signal transduction cascades. So again, this activated alpha subunit activating this phospholipase C, essentially causing real cellular changes. But something important to realize is once we have this activated alpha subunit with this GTP, we know as long as it's bounded to a GTP, it's activated and it can activate these effectors. However, these alpha subunits have intrinsic GTPase activity, which essentially means after a certain period of time, this GTP will hydrolyze into a GDP. And once it hydrolyzes into a GDP, now it's switched off. Now this alpha subunit is inactivated. Now that it's inactivated, now it'll go back to its original situation. And now it's back to this inactivated alpha subunit with the GDP, and now it's ready to do the whole process over again. As long as a new signaling molecule binds, now it can go through the whole process over again. So this is signal transduction cascades.